is Heidi Rehm, and I am the director of the Laboratory for Molecular Medicine at the Partners Center for Personalized Genetic Medicine. My laboratory focuses on translating new discoveries into clinical tests, as well as bringing new technologies into clinical use to make existing tests more accessible to patients. Before we were able to have access to next-generation sequencing, we did most of all our genetic sequencing by Sanger technologies. And this was a technology that was used for many years, very robust, the sort of gold standard, but it's not high throughput. And so we had a lot more technicians dedicated to sequencing, a lot less genes. And now with next-generation sequencing, that whole process has become much more efficient. We can use less staff and less resources to be able to sequence more DNA. One of the aspects of Illumina's technology that I think makes it most accessible is really the high quality nature of the sequencing technology. And especially for a clinical laboratory where we're not taking a research approach where we just take whatever we get. We have to sequence every gene to completion and ensure the highest level of accuracy of every base that's important to a patient. And so we choose Illumina technology because that technology gives us the most accurate sequencing that is critical for patient care. Illumina has been continually developing additional support for the entire workflow in the laboratory. Initially, their focus was in the instrumentation and the platform there, but now they have been developing reagents, the ability to make libraries, and really support both the upfront process that allows us to prep the samples, more efficiently combine them to reduce costs for patients, all up before they get loaded on the instrument, which is then what generates the actual sequencing data. So that whole process of supporting the part before it goes on the instrument has been a major area of focus, and that has been enabling more efficiencies and reduced costs for patients.